Okay, let's have a class. This will be our last lab section to do the practice. Thursday we'll have a review, then we'll have a break, and after that we have a final exam of lab. Because lab always is a week or two finished earlier than lecture, because we have to clean. You know, there's so many stuff there we have to clean. Okay, so what we're going to do today is analyze microorganism in food products. Okay, this is related to the food safety and food microbial safety. So we will talk about the detail in the lecture today. And here we're going to do some of the practice. Because it is not a BSL2 certified lab, so we cannot analyze pathogen here. What we will see is a total microbial population. Okay? Microbial population. And we will do numerous different type of the products, including tomatoes, hot dogs, ground beef, and chicken carcass. The key thing is that the preparing of these food products to numeration is different, is not the same. So we want to introduce to you one by one, okay? So first thing first is preparation. Number one, tomato. So first thing first we want to mention is that tomato, what we really care in the food safety standpoint is salmonella, especially in the Florida area. It's a number one tomato supply in the United States, and also Arizona, like Yuma area, and also the central area of the California. Those are the big area for supply. When you see the tomato like this is in the West Virginia local, maybe the high tunnel. It's not really traditional methods. What we really care about is this stain scar. This stain scar area in the center. Let's say you have a tomato. This is what we call it a stain scar area. This area is hiding lots of bacteria, especially this on the top. Okay? And the surface, of course, there is lots too. And there is a dump tank to processing tomato, and there is also regulation to talk about tomato, especially the concentration of the chlorine and the peroxyacetic acids. All those have requirements. So we will talk about some of them in the lecture. But here we want to show you how we process a sample of tomato. So I'll just give you an example. We have a nutrient broth, and we also have a cylinder. The cylinder is autoclaved. And we were using some of the sampling bags is like this. So how are we going to do the tomato? You tear it off using two hands to touch the linkage, and then you put the tomato in. Okay, now how are we going to do it then? We are planning to add, in this bag, we have a tomato there. We're going to planning to add approximately 150 ml nutrient broth. So that's 100, then we need to add 150. In order to save a little bit, let's add 100. Let's just add 100. Okay? 100 ml nutrient broth. So I'm using this. This is autoclaved. So I'm going to add 100. And then I'm going to dump it. Now, how are we going to do the sample? The idea is that we want to get all the bacteria out of this tomato surface. So how we should do? Like this. We're going to do a massage about 30 seconds. Then look at this bubble air inside. This is a very good. You can do shake 30 seconds. And then you massage again for 30 seconds. This will try to detach the microorganism on the surface. Okay? So will be 30 seconds massage, 30 seconds shake, and 30 seconds massage again. That will be the tomato, how we treat it. Okay? Now, we're going to do a second one, which is a hot dog. How are we going to do a hot dog? Hot dog is like this. So we have to see the hot dog. If you look at the ingredients, you look very carefully, you will see there is sodium phosphates, sodium diacetate. These two is actually antimicrobials. Because Listeria monocytogenes is a hot topic for the hot dogs. Monocytogenes. So, in the hot dog ingredients, there are some antimicrobials. If you look at the label, let's say sodium diacetate. And the tomato, by the way, we care about salmonella. Okay. Now, how are we going to analyze the bacteria on a hot dog? It is a little bit different. So how are we going to do? I'm going to get another bag. It's like this. Okay. Tear it off. I'm going to add one of the hot dog. Going to add in. And then the same thing, I'm going to add 100 ml of nutrient broth. So add inside. Now what we going to do? Look at the gas air bubble. The gap here is not enough to shake. So what we should do? Shake like this a couple times. The bubble is generated. Then you vigorously shake 30 seconds. Because tomato is large, you can directly do this is small. So vigorously shake 30 seconds. Okay? So what we have to process about this one, we usually say you have a hot dog in there. You have to do rolling and then shake 30 seconds. So that's a hot dog. Okay, what else we have? If you look at the products here, we had chicken and we have a ground beef. It looks like lots of food here. Okay, ground beef. And the chicken, we talk about chicken later on. So we talk about ground beef right now. What we should do for the ground beef? What we care about the ground beef, we'll mention in the lecture also, E. coli 0157H7 is a huge risk and zero tolerance. and it's zero tolerance. So when you look at here, there is a label tell you ground beef you cannot eat raw. You need well cooked. You look at the label here, they tell you to cook. Internal temperature 71.1 degree Celsius based on the research. This meat 85% lean, 15% fat. That tells you the ingredients, okay? And I don't think there is antimicrobials. Usually they come out of the supply chain, they do a radiation to find out like decontamination. So all we're gonna do is, so we're gonna tear off the top. Do we have a, sorry, do we have any of these scissors? If you don't have a scissor, we'll just use this, okay? For this one, I will use a small sample bag. The small sample bag is right here. And the same thing, to prepare, I bring chopsticks. So we make it a little bit easy. How much are we gonna add? Originally, we're supposed to be measured, but we'll just give to you is about 10 grams, okay? So what we should do is that we just get some, because otherwise it's going to be very nasty. So we get about 10 grams, and how much are we going to add there? We will be adding approximately 10 ml. Okay, so I'm going to add 10 ml. This is 20 ml, so I'm going to add 10 ml. Okay, then what we should do? We did it already. Stomach, yes. So if you are using this, you have to come here to do the stomach, okay? Like that. Stomach, about 15 to 30 seconds. I will do 30 seconds. And then you don't forget about, we have a two sides of the solution. Which one you should pick? Liquid side. So you always pick from the filtered side. So non-filtered side is the meat. You don't use the meat because it's gonna stuck there. Okay, so that's ground beef. The last one is a chicken. It's a big one, look at this. Chicken, how are we gonna do it? Okay, the last one will be a chicken. Chicken carcass. Okay, chicken carcass. We need to use a large bag. This is a large chicken bag. It's different because it's huge. Now what are we going to do then? Are we going to cut? No, we cannot cut the small pieces each one by one. So what we do? We're going to cut from the side. Okay. Be very careful. This you have to be two of you work together. This will be tear off. And then we'll try our best. Don't put the outside the bag there, but then you're gonna be gradually to dispense it inside. This is coming from Puduk Farms, by the way. The chicken is inside. Try to do this is not carry on the other bacteria. Now, what we are looking for the chicken, basically we are looking for Salmonella and Campylobacter. So how are we going to test those things? It's a big chicken there. How we do it? You're going to add in 400 ml nutrient broth. Some of the research also say 200 ml is enough. So we can try 200 ml today. And vigorously shake 30 seconds. Okay, how to do it? And we have another cylinder right here. This cylinder is about 250, so we need to use some of that. When you see here is about 250, so I will using this large one, and I will be dump about 200 ml, because 200 ml is a good resource, so I'll use 200 ml. It's enough amount. So 200 ml, because we can save some. So we add it. Okay, now this is called a vigorously shake, like this. Be careful about the legs and the wings. It's very easy to broke it. So when you guys do, I maybe gave you 2 bags. Okay? Somebody gonna ask me, look at the red color there. What are those? It's a blood, is that right? Is that good or not? Yes, it's good for numeration bacteria. This is some kind of, you're using a blood agar media in a blood solution. So that's a good thing. Lots of the time we are doing this, we shake, we want to taking all of them out. We got to testing this solution whether they have salmonella or campylobacter. And this is called a rinsate, or we call it a rinse solution. Okay? So this is how we do the chicken. So today will be your choice to do. I already prepared 4 samples. Some of you can use it. I don't want to waste it. It's too expensive. That chicken is like $15, $12 there. So I can do a demo for the next lab again, and you can use it. Now what we got to do then? We did this a couple of times. Okay, everybody, I will not say everybody, you can work together for 2 of you. But when you do spread plating, you're all going to do by yourself. 3 tubes. Each of them 9.9 ml. 9.9 ml. And 9.9 ml. All these solutions, liquid solutions, you are adding 0.1 ml from the first one, from the original one, go to the tube. Mix very well, then 0.1 ml go to the second one, mix very well, and then 0.1 ml go to the last one. We make a cereal dilution. So what is the dilution factor of the tube? 10 to the 2, 10 to the 4, and 10 to the 6. Then the same thing. We're going to add in 0.1 ml, which means 100 microliter onto the agar plates. Now don't forget these agar plates, you have to label your batch number, name, and the product's name. Okay, hot dog, just write HD. Tomato, TO. Chicken, write a C. Ground beef, write a GB, you know. And then, remember how we added it? Using 1 tip from high to lowest dilution. How we do the spread plating? This is 1 spreader from low to higher, high to lower dilution. Okay? So what is the final dilution factor? 10 to the 3, 10 to the 5, and 10 to the 7. I guarantee you there's a lots of bacteria there. Especially like ground beef, hot dog, and also tomatoes, especially tomatoes. Okay? This is a work you're gonna do today. Then Thursday we'll do the review. We also talk about the calculation. There is a lots of the little bit of math behind it because we have so many different products. Today you're gonna process the sample. I only have so many chicken, so the whole classroom can only have 3 people do chicken. I have millions of tomato, hot dog, and ground beef. You can pick any one you like. I also have these samples already. You can use them if some of you want to use. I don't want to waste them. Okay, it's not easy to get these samples. So go ahead to do it.
Dr. Shen, so, but everyone's doing